Good morning to all. I'm Wu Huan, representing the second affiliated hospital, Chongqing Medical University. Today, I will represent our insights on the construction of an excellent 7 mRNA-related gene model based on the cancer stem cells for predicting survival outcome of cervical cancer. A special thanks to our organizer for hosting us here in WCCRT 2023. Cervical cancer, abbreviated as CC, stands as a critical health challenge. CC remains a global burden on females' health in Africa, South America, Southeast Asia, and other countries. The incidence and mortality rates are highest, reaching 15.3 to 40.1% and 7.7 to 28.6% respectively. Consequently, discovering serviceable therapy targets and prognostic biomarkers for CC is highly necessary. Cancer stem cells are associated with adverse clinical outcomes of cancer. Cancer stem cells are a subpopulation of tumors, mainly responsible for tumor maintenance and spread. Cancer stem cells would produce many differentiated cells and highly value-added and self-renewal capacity, and we were the main components of tumor populations. Activation of cancer stem cells was known to lead to tumor recurrence and chemical therapy resistance. In recent years, sternness indices were conducted to profile sternness characteristics. Some mRNA expression based on sternness indices were associated with low-grade glioma and gastric cancer prognosis, providing new ideas for prognosis, recurrence, and metastasis predicting of gastric cancer and low-grade glioma. Next is materials and methods. First, data source and processing. The high throughput RNA sequence data and clinical information of CC were from TCGA database. The GSE 44001 and GSE 52903 datasets were from the GEO database as validation cohorts. The same treatment was done in the GSE 44001 and the GSE 52903 cohorts, and the 355 samples were retained respectively. Acquisition of sternness index based on the RNA sequence. Sternness index model trained from the progenitor cell biology consortium database with ORLC algorithm was applied for tumor sternness calculation. Indication of the mRNAsi-related genes. The Spearman correlation between each mRNAsi and the corresponding gene was calculated in TCGA CECS cohort. Consent consideration. The genes which had been found to be highly correlated with mRNA were used to perform the consent clustering. Patients were clustered into different sternness subtypes. The calculate distribution function and the consent matrix determined this optimal subgroup number. Sternness subtype comparison. Chi-square test was conducted to compare different clinical variables, combine TNG stage, grade, age, and sternness in the subtypes for difference exploration among the different sternness subtypes of TGEA databases. Difference in ceremonic landscape among the three subtypes were analyzed. DEG analysis and functional enrichment analysis. R package DML was used to calculate DEGs between the 31, 32, 33 group goal and KEGG functional enrichment analysis were performed on the DEGs in group C1, C2, and C3 using the R package customer file. Construction and verification of the sternness-related prognosis model. Cox regression analysis was performed with R package survival to select DEGs that had an impact on the OS of CC patients. K-curves were employed to compare the OS difference. The time ROC package was used to perform ROC analysis and calculate the AUC value to evaluate the prognostic ability of the risk score. Correlation between the risk score and the clinical characteristics. Evaluating the distribution of the risk score in the different clinical features, including TNM stage, grade, stage, and age, to assess the robustness and accuracy on the prognostic model. Survival differences were compared to the two groups stratified by clinical characteristics. GSEA enrichment analysis. We perform the GSEA enrichment analysis using the Fisher package, and the cluster profile package was utilized to functional annotation. Determinal of the tumor infiltration immune cells. We download 29 TME signatures from previous studies that could predict immunotherapy response. Use our package GSVA to obtain the SSGSEA score. Prediction of treatment of response. To verify the predictive value of risk score in immunotherapy response, we performed validation in the IMvigor 210 cohort. Next is the results. MRI is associated with cervical cancer's prognosis. Combined with the results, no significant difference in MRI was found between the different subgroups of TNM stage and age, and the MRIs for the CESC patients in the G2 and G3 subgroups was significantly higher than the G1. Look at the picture. Higher MRI means poor OS in both early and advanced stage patients. The results suggested that MRI based on the CSCs was closely related to their prognosis in CECS. Look at the figure. Molecular subtypes of CECS based on the MRI's SIJs. In the TCGA CESC cohort, distribution of various clinical features was analyzed in C1 to C3 subtypes defined in this study to see if there is any differences. We found that TNM stage and age did not differ significantly in the subtypes, but higher sternness features could be clearly observed in patients with C1 and C2 subtypes. Associations between molecular subtypes and genomic landscapes. To explore the differences in genomic landscape between sternness subtypes, we found that C3 subtypes show higher levels of TMB and puberty, while the C1 subtype show higher levels of pallidity. Meanwhile, we find that the majority of patients with the C3 subtype belong to the C2 subtypes with IFN dominant characteristics. Immunological microenvironments in certain types of CESC, immune cells activity and pathway activity related to tumor genesis and progression were higher in the TME or C1 subtype patient, while may also be the reason for the shorter OS in C1 subtype patients. Differences in immunotherapy between molecular subtypes. We found higher mDSA-CAF exclusion and tide score in the C1 subtype. This research indicates that patients with the C3 subtype might respond more favorable to immunotherapy. Identification of DEGs based on the different subtypes. DEGs in C1 group were mainly enriched in CAF and EMT-related pathways, a result consistent with the TME trend in this study. Look at this figure. Development and validation of the prognostic risk score. Survival analysis shows that patients with low risk had better OS than those with high risk. The prediction of 1, 3, 5-year survival probabilities indicates that risk score still has excellent AUC values, demonstrating that risk score has excellent performance in assessment of the prognosis of patients suffering from CC. Clinical correlating analysis of the prognostic risk score. Risk score construct based on the prognostic risk related MRI features of the DEGs in the subtypes had good robustness. Characteristics of immune inflammation pathway between the risk score groups. The relationship between risk score and the pathway activity score have found a significant positive correction between risk score and agnosis-related pathway. Explorations of clinical applications for risk score. The results show that the T-cell inflamed GEP score and Th1 IF1 gene signature SSGSEA score were significantly increased in the low-risk group, based on the R package of the agnomia. We analyzed the IC15s of 6 chemical therapies used in CC treatment and predict 3 significant correlation between R score and drug sensitivities. The results suggest that R risks may also serve a predictor of immunotherapy. Discussion. This study first explored the relationship between external characteristics and tumor prognosis in TME in CC. Identified 3 molecular subtypes including C1, C2, C3, and constructed a clinical prediction model for predicting prognostic and immunotherapy response. We found for the first time 3 STIR-related subtypes of CC, which provides new insights into the pathological subtype of CC. In the study, a clinical prediction model composed of SPRY4, PP1R14A, MT1A, DES, SEZ6L2, SLC22A3, and CXCL8 genes are constructed. SPRY4 may function as an anti-tumor gene in CRC, and SPRY4 suppresses CRC progression by inhibiting EZH2. In a word, our study identified stubbornness-associated genes associated with prognosis in the TGA public database and further constructed a 7-gene prognosis signature with promising ability in predicting survival outcomes and immune therapy response in the CC patient. However, more systematic and in-depth studies in the wet trials are needed to exclude the association. For the conclusion, we utilize mRNA-related gene for CSC features to develop new molecular subtypes of stubbornness signature in CC, which might expect to complement the existing pathological typing and providing insights into the mechanism of metastasis and drug resistance in CC. Our research also developed a 7-gene prognosis signature for CC, and there were promising clinical benefits of risk score in predicting prognosis and treatment benefits in CC. Acknowledgements. The study was funded by the General Program of Chongqing Nature Science Foundation. We will continue our study on the cervical cancer. Thank you very much.